discuss. And the important feature of natural products is a very strong dependence of the activity on the structure. So if we isolate natural structural congeners of known compounds, and we can see how their structural difference affect antibiotic properties, it lays a very good basis for further semisynthetic drug development. I will further illustrate those principles we formulated on some examples of our works on natural products. The first one is a novel class of glycosylated lipopeptides. We call them gauzyamycins. Those peptides were isolated from Streptomyces. Their structural novelty was striking. The peptides were containing previously unprecedented amino acids, and even the difference between two main components was very unusual because they differed on the side chain beta-alanine. Comparing gauzyamycin with known natural lipoglycopeptides, we can see that they differ very significantly in the peptide part of the molecule. The peptide core somewhat resembles anionic lipopeptides, but of course they are not glycosylated, and also there are crucial differences such as long exocyclic fragment, unusual deamino acid places, and the lack of calcium binding motif. Inspired by this striking structural novelty of gauzyamycin, we decided to elucidate its biosynthesis path. This slide is very small, so no one can be seen there, I understand, but the most interesting was the biosynthesis of unprecedented amino acid, amino-hydroxyphenylbutyric acid. And this analysis allowed us to assume that it was biosynthesized from phenylalanine via intermediate formation of gomophenylalanine. And to support this assumption, we fed our producer with fluorinated phenylalanine, and as a result, we observed the introduction of phenylalanine into the molecules of gauzyamycin, and MS fragmentation showed that fluorine atom was introduced into amino-hydroxyphenylbutyric acid unit. Why this result is interesting? Because even knowing the biosynthetic origin of the molecule, even using such an easy and straightforward technique as feeding the producer with non-natural amino acids, we managed to expand the chemical space of these peptides. And if we manipulate the synthetic gene clusters with strict understanding what we are going to achieve, this opens up a really big opportunities for expansion of the structures and production of non-natural antibiotics based on natural ones. That's why biosynthetic origin description is so important in case of natural products. Later on, very recently, we isolated several congeners of gauzyamycin. Gauzyamycin C was found to be N-methylated, and there were 3 analogs lacking carbohydrate moiety. That differences are not so significant. So the main component is gauzyamycin A, and gauzyamycin B contains side chain beta-alanine, and gauzyamycin C is N-methylated in this beta-alanine. So the most important thing about natural or non-natural, but any antibiotics, is its biological activity. So what about biological activity? First of all, to assess the mode of action of these compounds, we performed a bacterial cytological profiling. And surprisingly, we saw that gauzyamycin bring severe membrane damage and not inhibit cell wall biosynthesis like known lipopeptide antibiotics. When we compared kinetics of membrane activity of gauzyamycin, it was found to be similar to daptomycin and not classical pore-forming compounds like nisin. Daptomycin has a very complex membrane-associated mode of action, and we are sure that our mechanism is somewhat similar to daptomycin but different from daptomycin because our compound has narrow spectrum of activity. It inhibits staphylococci and it is almost inactive against enterococci. That's unusual for membrane-active compounds. And returning to the structure-activity relationship in this family, we can see that those not very significant differences between the gaussomycins led to dramatical change in their antimicrobial properties, leading to gaussomycin F that was practically inactive. And studying these biological properties, we noticed that gaussomycin activity depends strongly on calcium ion concentration, but this effect is observed only at high concentration of calcium. Then we studied the association of gaussomycin with the model membrane using NMR techniques, and we can say that gaussomycins introduce readily into the membranes. So summarizing, I won't go into any scientific detail, but the idea is that we are quite sure that gaussomycin have membrane-associated mechanisms of action different from any known compounds. So the natural products can still be origin of novel targets and novel modes of action. And probably gaussomycins, as they are not suitable for clinical introduction, but they can, if we further investigate the mechanism, these mechanisms can lay a basis for the novel drug development. And the last short example of the importance of the minimal structural changes for the activity of natural antibiotics. Here we studied the family of aromatic polyketides, tetracenomycin. Tetracenomycin X was described as a potent translation inhibitor, and we isolated two natural congeners. The first one was 6-hydroxytetracenomycin X, and when we tested it on the cells, on the in vitro, we found reduced antibacterial activity. But when we tested the inhibition of the ribosome by this compound, it was just as active as the parent compound. So these structural differences lead to a bad transport into cells with the same target affinity. And when we isolated the tetracenomycin isomer with the same molecular mass, it demonstrated no activity against the cells and no translation inhibition. And in silico analysis of its binding to the ribosome revealed that this hydroxy group that was methylated in this isomer is crucial for ribosome binding. So, to conclude, the main ideas I wanted to illustrate with my recent works is that the development of novel antibiotics is in significant extent problematic because of commercialization problems, but natural products are still a good source of new drug leads. And crucial in these studies are understanding of modes of action because they lay basis for further drug development. Thank you for your attention.
Thank you so much. Now we can take one question. Anybody related to antibiotic? Yes, Pawan sir?
Thank you very much. Very nice work, and that gives ideas for the future exploration. People have tried various ecosystems in search of the antibiotics, aquatic ecosystem as well as the terrestrial ecosystem. So the question always is that, let's say animals, in their skin they have got this property of killing microbes and so on. There's a huge range of that. You addressed part of it already in your talk. If you have to go to a new ecosystem in search of antibiotics, what would that be? Where there's a possibility of getting newer forms of antibiotics. Because anyway, we don't know much about nature. Maybe a few percentage, that's all.
There is a lot of studies, and of course, studying new ecosystems is not one answer for the question. But for me, the most interesting is oral microbiota of mammals, because I think that there are a lot of works on the intestine and of mammals and symbiotic bacteria there, but oral microbiome is less explored, and that's the point, that's the key place when the animal contacts the nature, and that's the place when symbiotic microorganisms might be found.
That's very interesting. Thank you very much.
Thank you so much, Dr. Vera Alferova. Your explanation related to ecosystem of antibiotic. You already explained like microbiological, molecular biological, and like chemistry biological, antibiotic, new approaches to be done, and your explanation related to gossamicin is really, really superb. Thank you so much. And now I would like to invite Dr. Soumyanand Chakrabarty, ICMR National Institute of Malaria Research, Bharat. His title is on anti-malarial delivery with ferritin-based protein case, a step towards developing smart therapeutics against malaria. Dr. Soumyanand Chakrabarty, a structural biologist and biochemist, is currently engaged in research at the National Institute of Malaria Research, Bharat. His primary focus lies in developing therapeutics for infectious diseases such as malaria. With a postdoctoral background from prestigious institution in the USA and Europe, he has demonstrated exceptionally productive with 53 publications generating 2,365 citations and an H index of 27. A big round of applause he is expecting. Now I hand over the mic and the stage to Dr.
वीडियो
Soumyanand
चलाने के
Chakrabarty.
लिए वीडियो है तो एक लेजर कौन सा है? Thank you, organizer, for giving me this opportunity to present my work here. So basically, I am working for Protein Cage for almost 10 years now. So, but I am in the Institute of Malaria. So I'll go to that first, means what I am doing. So basically, this is kind of last 5 years work that I did with the structural biology. But then in 2020, I joined this institute, National Institute of Malaria Research. So where the mandate is malaria, and also government wants to eradicate malaria from India in 2027. So the mandate is we have to do something with the either parasite or with mosquito. So with my background in Protein Cage, I thought of opening a new stuff. So basically we are doing two parallel projects in my laboratory. One is protein cage-based therapeutic application, one is drug delivery, and also I am trying to do antigen, trying to use protein cage as a scaffold for antigen display, as well as I am also working in the understanding biochemistry and means drug designing mostly against mosquitoes. So I'll come to that, means today I'll talk about my means last 10 years work about the protein cage based therapeutics and how to generate protein cages. However, before going to that, I just wanted to show you what kind of work we are doing in the mosquitoes. So because this is a Bill Gates notes published in 2015, you can clearly see the mosquitoes are the most deadliest animals in the earth, and they killed more than, means each year they are killing more than million people actually. And if you look at the statistics in 2021 also, so malaria plus dengue causes more than 500 millions of infection. It's not that there is no insecticide available to treat mosquitoes. There are certain, and the latest developed insecticide is pyrethroids, which developed in 1970s. However, the problem is last 60 years there is almost no insecticides developed, and as a result there are a lot of resistance, and India is Indian mosquitoes are showing resistance against all these insecticides. So that is a cause of concern. So we need to develop new insecticides, and that is something we are trying to understand. We are trying to understand different important or essential biological pathways. One of the drawbacks that we are having in the mosquito field was that we don't have a very good draft genome present, but in 2016 and onwards we have mosquito draft genome, so it would be easy to understand their structure and target them. So in my laboratory we are mainly focusing three pathways actually, so we are trying to understand how mosquito recognize odors. So basically, so how mosquito recognize the odor. So, and there are a lot of protein actually involved in the mosquito odor recognition process, and we really don't know about that. And in insect, it's really complex, and at least 200 proteins are involved. So we are trying to understand it from odorant binding protein perspective, but in odorant binding protein also there are almost 70 proteins in mosquitoes, so we are trying to understand it. Bhuvan my postdoc Bhuvan is having a poster there, so if you are interested about how mosquito actually processing odors, you can, he will have a talk day after tomorrow, so please visit him. Otherwise, we are also looking at the mosquito is having a huge stress response actually, because recently Indian mosquito invade Africa, so mosquito can survive in many different climates. So we are trying to understand how mosquito can survive in such a stress condition, and that is the reason we are mainly interested on heat shock proteins, because there are and among heat shock there are many heat shock protein families available, but we are interested 40, 70 and 90 at this moment. And Bharti is having a world talk again day after tomorrow, so if you're interested more about mosquito heat shock protein, you can talk to her. And also we are looking at iron transport, because mosquito is consuming quite a lot of blood, and we don't know what is the fate of the blood and how they are utilizing this blood. So, and we are working with a molecule called ferritin, which I'll talk about today also, but we'll talk more about its drug delivery application. But ferritin can be very important molecule for the iron transport in mosquito, and Tupe will present that in day after tomorrow. Okay, so that is our mosquito research, but today's topic is completely different, so I am changing my gear and talking about this protein cage. So if you think about a protein cage, the biggest inspiration is a virus. And it looks like this. So this is basically a protein cage, ferritin cage. So inside is a hollow cavity is there, and outside you can do a lot of decorations. Nowadays you can, because of the advent of the bioconjugation techniques, you can put antibody, you can put aptamer, you can put nanobody very easily. And inside people have shown that you can put nanoparticles, enzyme, antigen, contrast agent. So, and this kind of structure is very good for electron microscopy because of the symmetry. So you can get a very high resolution structure out of it. So I'll show you that different type of cage that you, So protein cage can be differentiated into three categories. One is VLP, means virus-like protein cages. So if you think about viruses, and if you take out the nuclear material out of it, then what you will get is the cage. So these are the biggest family of protein cage that you can get. Then the next is the bacterial-based protein cage. So in bacterial-based protein cage also, there are a lot of bacterial microcompartment you are having, you can take them and you can think of, means you can use them for a synthetic delivery purpose. And last but not, means the most recent one is the synthetic protein cage, where you can engineer protein interface and generate your own protein cage. The beauty of this synthetic protein cage is that you can control their assembly and disassembly very easily. And again, so you can do cage modification in different way. You can do inside modification, outside modification, and even interface modification, and you can generate multifunctional protein cages. Okay, so I'll first start with synthetic protein cage generation, which we did it in Poland with Jonathan. So this is basically from the synthetic. So this is not, you cannot find it in nature. So this is a ring-shaped protein actually. So basically this is a transcriptional factor available in the bacteria. And so what we did actually, we did a modification, a cysteine modification in its surface. And when we did the cysteine modification on its surface and then incubated with the gold nanoparticles, we get this kind of beautiful cages as I shown you before. And so the condition for this cage is that when you have this mutation on the surface, that is K35C, and the gold nanoparticles, only in these two conditions you can get the cage. In rest of the condition you cannot. So we are trying to understand how gold is actually accelerating this cage formation or not. And okay, so there is a video next. So could you please play the video? Okay. Yeah, so this is the cage, how it looks like. This is a cryo-electron microscopy of the one of the biggest cage that we have, and in few minutes when you can clearly see that the beauty of these cages lies in the two cysteine residues are getting stitched by a gold atom. So this is one of the largest protein cage available so far, and this is an inducible synthetic cage from using gold nanoparticles. So, and another good thing about this cage is that it's extremely stable. You can boil it, it will still not break. Means the melting temperature is 116 degree. So, okay, so these are the basically the stability work that we are doing with these traps, and we found that this cage is extremely stable in different pH condition, different temperature condition. Even with the different chaotropic agent, we found that these cages are extremely stable. And we did a lot of electron microscopy to show that, means this cage is stable. Okay, so now, but I told you earlier that this cage is formed by the cysteine gold cysteine bond. So basically, in reducing condition, this cage is actually disassembled. For example, if you put DTT, so we have a trigger for this cage. For example, if you put DTT or T-shape, this cage getting completely disassembled. However, if you put an oxidized version of the GSH, GSSG, this cage is stable. So although this cage is extremely stable at different temperature and pH condition, but it has a trigger. Trigger is redox, and if you put redox, it will be easily disassembled. So with that cage, next, this is an example of high-speed AFM where you can see that how cage is disassembled in presence of DTT actually. This cage is already kind of broken, and now the subunits are coming out, and eventually this is completely disassembled. So this is a direct demonstration that DTT can disassemble this cage, means redox can disassemble the cage very easily. Okay, so first I showed you that how to generate a protein cage from scratch protein molecules, and now I'll change the gear. I'll go to another protein cage, and I'll talk about cargo encapsulation, because as we are interested in drug encapsulation, so cargo encapsulation is kind of our interest. So for that, we'll take a different protein cage, and that cage is ferritin. Ferritin is available almost all human, means almost all it's even available in human and other organism except few yeast and plants. But we discovered a ferritin from Thermotoga maritima. This is a bacterial ferritin. The interesting part of this ferritin is that this ferritin doesn't exist as a cage, it exists as a dimer actually. And if you put salt into it, then it will form the cage. And so we did a lot of experiments, mainly chromatographic based assay, to prove that what is the best condition for this cage to form. Because normally if you have a cage like structure, then especially with the ferritin like cage, it's very difficult to disassemble. You need very low pH to disassemble it. So however, in this cage, in this protein, we have a metal protein trigger, and the ferritin, the physiological function is to store the iron. So basically the inside lumen is the negatively charged. So now an iron is a positively charged, so basically there is an electrostatic interaction going on so that iron can go inside it and forms the nanoparticles. And as I told you, ferritin can be found in everywhere, so from bacteria to human, ferritin is there. So using this unique system, we thought, why don't we do some protein encapsulation or not. I'll tell you what is the advantages of doing protein encapsulation inside it. So, but before going to that, I just wanted to introduce this concept of protein supercharging. So basically protein supercharging, you are actually modifying a protein surface to either highly positive or highly negative, so that you can, trying to force a protein for more electrostatic interaction. So this concept was not new, it was first introduced by David Liu of Harvard. What he showed that he made, I think all of you are aware of green fluorescence protein, but when he made this minus 30 and plus 36 variant of GFP, and then when he heated it 100-degree temperature and cooled it down, you can clearly see that in the normal GFP it's getting aggregated. However, in the minus 30 and plus 36, both are extreme positive and negative charge supercharged GFP, they are actually getting back their color. It's because they have a highly charged, so they repulse each other, and as a result, although there is a transient aggregation is forming, but with time they can get back to their own color. We use this for a different purpose. I told you already that ferritin inside surface is negatively charged, and to put something very strongly inside it, we need something very positive. So what we did actually, we incubated and another reason we took GFP is that it's very easy to track, because if you have GFP inside a protein cage, you can easily see that. So we just incubated these two together in presence of metal, and we can, because of the projector, I cannot show you, but we did quite a lot of assay to prove that the GFP is inside, and you can at least incorporate 3 to 4 GFP molecules inside this cavity of the ferritin. However, when we sent this paper to the nanolators, they asked us, okay, you put a protein, that is fine, but you should show enzyme encapsulation inside it and some of the application of it. So first of all, we put an enzyme which is having almost same isoelectric point as GFP, and when we put this and when we looked at the literature, we found lysozyme is having the similar isoelectric point. So the surface charge of lysozyme almost similar to GFP, and when we put it lysozyme, we found few interesting stuff. So first of all, it is protected from the protease, which is kind of expected because there is a ferritin shell is there that is protecting the enzyme, that is fine. But also when we are heating the enzyme, we found that the enzymatic activity remain even at 80-degree temperature. But normal protein is losing its activity within 80 degree. So this is mostly because of, we believe we can create a crowded environment inside. So this is kind of conformation locked environment. So we believe there is a high activity of protein even at higher temperature. So, sorry. Okay. So in the next, we are actually in the contact with a guy called Maury. Maury is actually famous for protein cage-based material synthesis. So when we went to him, he actually suggested us, why don't you make a material, because you put a antimicrobial protein inside it, why don't you make a material? And he has some gold nanoparticles. We incubated that and we get this face-centered cubic-like structure, means as you can see here, and this is enzymatically active. So this is so if you look at we started from a few nanometer of protein and we make this massive macromolecular structure which is enzymatically active. This could be applied in many biological application. So with that, I showed you first how to generate protein cages, next how to encapsulate cargo inside it. Now I'll move to some of interface engineering. As you can see from here that you can do a lot of stuff with protein cages. So one of them you can do interface engineering. And if you remember about these protein cages, that these protein cages are actually assembled in presence of salt. So we try to understand how this assembly is possible and whether by interface engineering can you make a stable cage which doesn't need a metal for its assembly. When we carefully looked at the structure, we found that there is a big repulsion going on between two residues, glutamic acid residues actually, that you can see here. And when we try to do molecular modeling and trying to change the interaction, we found that there is a favorable attraction that can be possible. And this repulsion actually, metal ion is stabilizing that. So with that in mind, we actually make several mutations in the E65 position. We almost generated all the possible mutation that one can perform in the E65. And as per our expectation, when we found that when we are changing the charge completely, you don't need a metal ion for its assembly. It gets assembled on its own. However, if you have the repulsive interaction going on, because then only you need a metal ion for that. So with that shot that we did the engineering to show that this cage assembly is highly tunable, and you can also put enzyme inside it, and based on your application you can use it for different purposes. Okay, so this is all this showing that when you have different mutants, what will be the, so when you have a E65A mutant, you need a nominal salt. When you have this E65D and Q, then you have magnesium to assemble it, and when you have this E65R or lysine or arginine, you don't need a salt for their assembly, they will form the assembly. So with that, I am talking about so far all things except malaria, how we can use this kind of cage for the application of malaria. I am coming to that actually. So ferritin as a drug delivery vehicle is not new. The ferritin was discovered in 1937. Sorry. And even in 1960s, it's known that you can incorporate inside the cell. And recently, in last 10 years, there are a lot of publications shows that it's really good delivery vehicles for the cancer cells. Some of the ferritin drug conjugates, especially with the doxorubicin and paclitaxel, are already in the clinical trial, and the first round of clinical trial showing very good result. And they are better than the antibody drug conjugate in thermostability and drug loading ability and ease of production. So because ferritin is very stable. So with that in mind, we started doing, because I am in an institute, National Malaria Institute, our mandate is malaria, so we start putting antimalarial inside it. And and we are quite successful in putting several antimalarial. We have tried 10 antimalarial, and out of 10 we are able to encapsulate 8 of them. And with chloroquine, we have seen quite a good result. It's the toxicity means it's producing almost same as the means the normal molecule. So you can encapsulate antimalarial molecule inside it. And the good thing about it that ferritin has almost no toxicity and very little immunogenicity. And immunogenicity also you can change it based on your decoration strategy. Second is RBC is full of transferrin receptor which ferritin can use for getting internalized. Also ferritin recently shown it can cross blood-brain barriers, so it can be useful for the cerebral malaria, which is the most fatal form of malaria. In fact, 2019 one group showed that when they just injected the mice with the ferritin and they found pretty good protection of the endothelial cells from ROS-mediated damage actually. And so according to them, ferritin can be used for the cerebral malaria. So we are working in this line, and we recently submitted this paper. It's under revision in biochemistry, where we encapsulated all these drugs, and Cherish, Shruti and Tupe did all this work. So, okay. So before going for my final slide, I just wanted to show that these protein cage are really getting a lot of attention these days because to the scientist community because of its multivalent display. Even the malaria vax that is used, or the RTSS, it's use a protein cage-based scaffold for displaying CSP proteins. So and some of them are also in the clinical trial, for example, RBD. So because of the multivalency and the high stability, protein cage-based antigen display can have very good immune generation actually. So with that, thank you. We are a part of a big group of Dr. Kailash Pandey, and these are my students. They all, most of them having a oral presentations, and Shelly is having a poster presentation. So if you are interested in more of my work, you can visit them. And also I got, so the work starts in Poland, so some of the funding from the Polish agency, and I am also funded by the Ramalinga Shami Fellowship and ICMR intramural grant. And thank you all my collaborators, because this amount of work would not be possible with their help. I am happy to take any question if you have.
Thank you. Hello. Thank you, Dr. Soumyanand Chakravarti. Now we can take questions. Only one question can be asked because we are running short of time and everybody is waiting for the lunch. Na?